Greetings to the esteemed review panel, our mentors and teachers. This is Capstone Team 18, Electronics and Communication Engineering. My name is Jeeya Prakash, a final year ECE undergraduate, 10185 6009.
My name is Anshumaan Singh from ECE 101806070.
My name is Dhananjay Raina. I am from ECE department and my roll number is 101806051.
My name is Prashant. I'm also from ECE department. The roll number 101806208.
My name is Shreyansh Soni. I'm from ECE department and my roll number is 101806057.
Our project is called Brain Tumor Detection in Medical Images using image processing and neural networks. We have tried to construct a denoising mechanism by developing our very own denoising filter from scratch for salt and pepper noise present in MRI images, which is one of the many dominating types of noises because of the presence of which reading of clinical images becomes difficult. Further, with the help of convolution neural networks and machine learning, we have designed a detection algorithm that checks for tumors in an image. Our inspiration for this project comes from the very fact that because the tumors are so benign, the presence of noise makes it almost impossible for a very experienced doctor to read and catch a tumor. This causes the cancer cells to grow undetected and metastasize in the body, and it is only detected when it is too late for the person. Therefore, we have tried to come up with a more efficient denoising algorithm so that the quality of an MRI image can be improved. The working of our project takes place in two parts, the denoising section and the detection section.
In order to learn about denoising of images, especially salt and pepper noise, it was quintessential that we read about the existing techniques and methodologies in the domain. Some of the filters are a new adaptive switching median filter or AWSM filter, DEMF filter, SWMF filter, etc. After reading multiple articles of various filters many times and implementing them, we came up with our own idea, which gave better results than the previous filters. From the chart shown, we can clearly see that our filter in brown gives a better peak signal to noise ratio than the other filters. In image enhancement factor as well, our proposed filter reigns supreme. When it comes to CPU time, our proposed filter runs on par with PSMF and FSB MMF filters, but due to better PSNR and IEF results, we can conclude that our overall filter is more efficient than the others.
So in order to create a new filter, we first looked for a basic filter idea which can act as a backbone for our filter. In our case, after an extensive literature review, we decided to go with the median filter due to the simplicity and accuracy over the existing mean and recursive or interpolation filters. Then we decided to attack some of the problems these filters were facing. We recognized that even though median filters were accurate, they weren't fast enough as some of the mean filters. Our filter, first of all, takes an input image and it generates a flag mask. The flag mask is basically telling us in the image where a pixel is noisy and where it is not noisy. So if a pixel is noisy, which is if it's obtaining a value of 0 or 255, it will flag it 0. And if it's a healthy pixel, we will directly give it to the pixel array. Then after that, we will set our window sizes to 3 as the minimum and a maximum window size of 7. We will now look for number of healthy pixels in those windows and keep on increasing our window sizes as we keep on going. As soon as we find some healthy pixels in our window, we will take the median of those pixels and replace them in the pixel array. In case we don't find any healthy pixel till we have reached the maximum window size, we look for different conditions and we look for pixels in already the pixel array we have formed. That is the processed pixels that we have formed from the noisy image. Now I will show you the working of this filter on our MATLAB setup. Now, as you can see, this is the original image which was fed to the algorithm. And this is the noisy image which was fed to the algorithm for denoising. And this is the image we got after the image was denoised. So you can see that this is what the actual image looked like. And this is what we recovered from this image. So we can see that our algorithm not only was able to get much of the original details from the image, it also did it in a very less amount of time. Moving on to the next slide is the detection of the tumor in denoised images.
So for image detection through machine learning, we have first acquired a dataset through BRATIAS 2017 and BRATIAS 2018. The BRATIAS 2017 dataset comprises of pictures that have been gathered from 285 glioma patients, out of which 210 were HGG cases and the rest belong to the LGG cases. We have tried to distinguish between different types of tumors such as glioma tumors, meningioma tumors, and pitchbury tumors. For image segmentation, we have used the architecture of UNIT, which is very efficient in terms of performance. The unit architecture has a basic U-symmetrical structure where left of the U structure provides us with an encoder and the right side of the architecture helps us to perform the decoding task. This architecture is able to concatenate two different layers, namely the encoder and the decoder, which eventually allows the feature map to have both low-level and high-level features.
Moving on to the classification section, the use of convolutional neural networks is immense when it comes to examining of medical images. So here is the demonstration of our working model. Here in prince.py is the Python script of our model. Let us upload a sample image. So as you can see here are the results where we can see where the tumor is present. So this is using the help of the semantic image segmentation, and here you can see the type of tumor it shows with the probability of 86.5%. So more specifically, the goal of semantic segmentation here in this model is to label each pixel of an image with the corresponding class of what is being represented. In our case, each pixel is either a pixel having a tumor, which is shown in yellow in the output, or a pixel having no tumor, which is shown in violet.
So after we were able to create our machine learning model and denoising model, what we did was we fed all this noise to our images, as you can see in this table, and all this noise was fed to the images, and the model was trained. And again, the images were denoised and the model was trained again. And then we recorded all these results over different densities of the noise. So as you can see, the noise was 10%. Over that, the noisy section, we can see the intersection over union is 0.95. Whereas when the same images were denoised, the model showed a result of 0.98. Same goes with dice coefficient. So as you can see, the denoise section is performing significantly well when compared to the noisy section, and the results are even more jarring after 50% noise has been introduced in the images. The noisy section just keeps on degrading at a very higher rate, whereas when the denoised images are used on the same model, the image quality degradation is not so much, and hence the model is able to predict better.
We also take a lot of pride in sharing that a research paper written by us under the guidance of Dr. Bharat Garg and Dr. Rana Pratap Yadav on the algorithm that was devised for this project has been accepted at the International Conference in Artificial Intelligence and Big Data in Digital Era, Vietnam. Thank you.